Hello, my name is Alessandra Zacchio, and today I will be taking you through the steps of the cell cycle. Before we begin, we must know what the cell cycle is and why it is important for our bodies. The cell cycle is the series of events and stages a cell undergoes to grow and divide. It is important for our body cells to grow and divide so that we too may grow and develop. Having multiple smaller cells in our body also makes transporting materials between cells easier and more efficient. The first stage of the cell cycle is interphase. The interphase is the longest stage. The first step of interphase is known as G1. In G1, the cell grows and produces some of the necessary organelles for dividing. In this diagram, we can see the cell membrane that controls what goes in and out of the cell, the cytoplasm that holds the cell's shape and organelles, the nucleus, which stores the DNA, making it known as the control center of the cell, and finally, the DNA that provides the cell instructions on how to grow, reproduce, and perform other necessary cellular functions. During the G1 phase, the DNA remains tangled. The next phase of interphase is known as the S phase. In S phase, DNA replicates for cell division. It is important that cells replicate their DNA so that the two daughter cells that will be produced will have the necessary genetic information. The final stage of the interphase is called G2. In G2, the cell makes organelles such as centrioles and spindle fibers. The cell continues to grow in preparation for cell division. The centrioles are tubes of proteins that make up the cytoskeleton. Spindle fibers are tubes of protein that make up the cytoskeleton. They both play an important role in cell division, specifically in the dividing of chromosomes. Before we enter the prophase, we must discuss what chromosomes are, as it helps us to know how cell division works. Chromosomes are structures of condensed DNA wrapped around proteins called histones. Chromosomes are made of two sister chromatids attached by a centromere. The first stage of mitosis is known as the prophase. In prophase, the nuclear envelope, or the nucleus's membrane, begins to break. The DNA that was once inside the nucleus condenses into chromosomes. Centrioles then move to their pools. Next is the metaphase. During metaphase, the centromeres of the chromosomes line up in the middle of the cell. Two spindle fibers from each side of the cell connect to the centromere of each chromosome. In anaphase, chromosomes are pulled by their spindle fibers to opposite sides of the cell as individual chromosomes. The microtubules then start to disassemble. In telophase, chromosomes start to tangle from their chromosome formation into tangled chromatin. The nuclear envelope forms again and the nucleus starts to become visible. The final step, cytokinesis, occurs in two different ways, one in animal cells and the other in plant cells. In animal cells, the cell membrane is drawn to the middle of the cell and pinches two new identical daughter cells. The cell creates an actin ring, a cleavage furrow, and a nucleus. The cleavage furrow is the small groove that is made between the new cells and the actin ring. It is the section of the membrane that divides the two new daughter cells. However, in plant cells, the cell wall is too firm to be pinched inward. Instead, the cell plate is created between the two nuclei and grows to infuse with the existing cell membrane. The cell wall divides the two new identical daughter cells. Now you have learned about the steps of the cell cycle. I hope it is now easier to understand why the cell cycle undergoes these steps in order for our body to grow. Thank you for listening.